So again, we're talking on the types of phospholipids. One is phosphoglycerols. So take note of the structure. In all glycerophospholipids or phosphoglycerides, so it's the same, huh? Phospholipids, phosphoglycerides, or glycerophospholipids the same. In all glycerophospholides, you have the lecithin, cephalins, and phosphatidylinositols. The fatty acids and carbon 2 of glycerol is always unsaturated. Kani. So in all of the glycerophospholipids, the second fatty acids here is always unsaturated. You can notice here. The first one can be saturated, easily saturated, but the second one is always unsaturated. That's what they observe. Now you have here. And of course you have your head group here. So if the other alcohol is choline, like this one, you have your choline here, a quaternary ammonium compound. The glycerophospholipid are called phosphatidylcholine. Okay, you noticed here that you have the head group substituent here. So here you can see here that it is also polar. You have here the amine group with 4 bonds here. So it is cationic. You have nitrogen bonded to 4 group of atoms. Cationic, so it would have a charge of positive here. So again, the structure, glycerol, CH2O, CH2O, but is esterified with fatty acids. You have your fatty acid here like stearic. The second one is an unsaturated fatty acid. That's what we mentioned. And CH2 instead of an ester, carbon ester here, you have your phosphoric ester, diester. You have an ester here, P double bond O, P double bond O. So it's a left and to right. It's a diester, phosphoric diester. And it's being bounded here. If it's a choline, you call this one a lecithin. Choline, no? So you have an alcohol. You notice here, you mentioned niya kaganina nga it's an alcohol kani. So you have your fatty acids, you have your phosphate, and you have your glycerol and alcohol. You can see here, you don't see an alcohol. Why is that? Because the choline, which is an alcohol, is already esterified here. You know, there's already some kind of reaction. You don't see already the alcohol. But when the alcohol, when it do reactions with phosphate to form an ester, na mawala si alcohol. It's one of the reactions if nakaabot mo sa reactions of esters with alcohols, no, like carboxylic acid and an alcohol, mawala ang alcohol. In the case of the forming a bond, no, instead you get an ester. Okay, pag may alcohol and you have here, mawala ang alcohol, but instead you get an ester. As H2OO, mabound siya diri. Okay, you have the phosphate. So again, you notice on the structure on the second carbon is unsaturated fatty acids, and you have here, if the X is lecithin, if it's choline, what is choline? You have CH2, it's a quaternary ammonium. Okay, and CH2. Yes. Nainangutan na? Yes. By the way, naitaga si DL2 diri nga nung gamay raman ang makakuan sa tong discussion. Tingala ko ni si DL2. Naitaga si DL2? Anyone? Anyone?
Yes.
Okay, so na-inform ka na. Okay, let's move on. So you have your lecithin as an example, and then you have also your phosphatidylcholine. So this is a typical lecithin molecule, has a stearic on one end and then a lake in the middle. You have your linoleic. So lecithin or phosphatidyl is an example of phosphatidylcholine. No? You have a phospholipid with a choline. So that's lecithin is a major component of the egg yolk, and you have other lecithin molecules that contain fatty acids, but the one on the end is always saturated and the one in the middle is always unsaturated. I've already mentioned this one. But an example of phosphatidylcholine, okay, wherein you have phosphatidylcholine. There is a general term for a lecithin, no? Ang lecithin is already called lecithin lang pag the first fatty acid is stearic, the second one is linoleic, and you have here the choline. But if kaning mag-vary na si fatty acid here, it's not stearic, it's not linoleic, another unsaturated fatty acid, that we generally call this one as phosphatidylcholine. And it's the common name lecithin here. But if you have here, okay, you have the major component, the lecithin is a major component of the egg yolk. And you notice, no? You notice in this is part, it can see this one in the egg yolk, that you have the non-polar part and you have this one is the polar part. Kani polar, put this one is all polar part. And you can see that your egg yolk, no, having a non-polar and a non-polar and a polar end, egg yolk is actually usually used as a surfactant, as a binder. For example, when you're cooking, if you're into baking, no, so ingredients in baking is that naamoy oil. Usually it's hydrophobic, no, usually oil, and then you have your water part, and then you have your kind of mga flour, right? So you want to bind them together, but of course, if they are just water and oil, dili naman na siya ma-bind, right? Because there's non-polar and polar mixture, right? So you need an emulsifier or surfactant, and that is the purpose of our lecithin or phosphatidylcholine, because we have the polar end and you have the hydrophobic end. One end, the hydrophobic end, will bind, if you are into baking, will bind into the oily part, and the polar end will bind into the polar parts sa inyong kuan. For example, if you're baking, no? And again, that's the reason why you have your lecithin as used as an emulsifier. And because of its polar and non-polar portion of molecules, an excellent emulsifier and is used in mayonnaise. And it is one for this reason that can have another activity in your laboratory, which is preparation of mayonnaise. No? And when you prepare your mayonnaise, there's the polar ingredients and usually non-polar ingredients. You want to mix them all, and that's why you have to add an emulsifier, and that is the purpose of using the egg yolk. Okay, pag magluto mo sa kusina and you want to bind all the ingredients nga medyo oily siya and there is a part nga medyo watery siya, no? Pag sila lang mabungkag na siya, there is no way that they will interact with each other, and that is the reason that you will have to add an emulsifier. Okay, so you can note that lecithin is a negatively charged phosphate group. As a negative charge to the phosphate, we have here one of the oxygens of phosphate. No, the singly bonded oxygen is negatively charged, and it is neutralized by some further parts for the whole molecule. No, it's neutralized by the lecithin molecule, which is positively charged. Sige, note that lecithin is a negatively charged phosphate group and a positively charged quaternary nitrogen from fluorine. These charged parts of the molecule provides a strongly hydrophilic head, whereas the rest of the molecule is hydrophobic. Okay, I have already mentioned that. When a phospholipid such as lecithin is part of a lipid bilayer of the hydrophobic tail, paying towards the middle of the bilayer and the hydrophilic end line, mo na siya, balik-balik nga istorya. Again, when they act as an emulsifier, okay, this is what they do. The non-polar part of the phospholipid will be situated next to each other, or either will be interacting more with each other. Okay, and again, so this is when you are looking into the lipid bilayer. Okay, kung sila lang, no other substances. But for instance, naamoy another molecule, another substances. For example, when I say theoretically, when you are baking, naapay mga oil and so on. So where do the oily part of your cake or ingredients of cake would go? So it would go somewhere here. Okay, where there is an interaction, hydrophobic interaction with the non-polar part of your lipid. And here, the exposed, which is the polar end here, where this is where the interaction with the polar ingredients of your ingredients sa cake, sa baking. For example, sa may mga polar, for example, flour, no, where it contains a lot of OH group. Isa pa, katong mga kung naamoy milk, okay, and you can also have here, okay. And pag walay other compounds, of course, you expect that the lipids will always form like a double layer or a bilayer, no? One layer, another layer, but the hydrophobic or tail will be interacted with each other, and the polar end will be again exposed to water. So there's another kind of a phospholipid. We call this one as a cephalin. Okay, you can see in the structure you have still the CH2, the glycerol, and the phosphate group. And in the glycerol, first and second carbon of the glycerol, you have your fatty acids, the bonded fatty acids. But what is attached to your phosphate group is not the choline, where you can find illicit in, but you have the ethanolamine. For this case, you call this one as a cephalin. So instead of choline, they contain another alcohol such as ethanolamine. You can see here again. You will ask, asa man nagiingon diri nga si phospholipids would contain fatty acids, glycerol, and alcohol. No, you don't see an alcohol in any part. Again, this one is derived from the alcohol. Before it bonds your phosphate here, this is actually CH2, NH2, and OH. When it binds here to the phosphate, dili mo makita ang COH because it's already bonded here. Okay. And of course, NH2 amine in water, dili mo na siya makitaan as NH2, but it is always protonated. So CnH2 would always react with water to form NH3+, so cationic siya here. And in fact, that's what you have learned when we talked about the amino acids. You don't see NH2 in water as NH2 because it will interact with water. You will have your NH3+ to discharge. No? So here is a cephalin when you are using ethanolamine. So this is your ethanolamine where you have your OH, binds to your phosphate group, dili naman makita ang COH, but then you will get an ester, but not the common ester acidyl bond O, but the phosphate ester, pidyl bond OO. Okay, this is how it binds. And the tail one's NH2, when it is in water, or yeah, you don't see NH2, but it is the protonated amine. And when it is protonated amine, you have the positive one. Okay, so you have here. And if you have, for example, serine attached to your phosphate group, then you call this one phosphatidylserine. This one is phosphatidyl ethanolamine. You call this one also cephalin. But in particular, you call it phosphatidyl ethanolamine. This one is phosphatidylserine. When instead of ethanolamine, you have the serine. Serine is an amino acid. If you remember, you have here C alpha carbon, C-double bond O, you have NH3, you have your hydrogen attached, you have CH2H. This is actually a serine, and this is chiral, of course. And so when you have here serine attached to your phosphate group, we call this one as phosphatidylserine. This is also called your cephalin. Okay, so this the other one is phosphatidylcholine when you have your choline. So there's another one you call as phosphatidylinositol. Your phosphatidylinositol is like the same structure, no? Acidyl bond. It doesn't matter kung unsa nga fatty acid will be added to your carbon 1 and carbon 2. But no, the easily on the carbon 2 is always unsaturated. Okay, so you have your CH2 and then phosphate. Okay, that's the difference of the third carbon of your phospholipids, right? But again, it would matter kung unsay maka-attach to your phosphate. Kung it is choline, then you have lecithin. And then when it is ethanolamine, you have cephalin, and whether it's ethanolamine or serine. But if you have this one, this is a sugar, no, which is more. it looks like a sugar, but not the real monosaccharide. Now there's no O here. This is actually inositol, no, the variation of your monosaccharide, but not really the monosaccharide. If you have the inositol, you call this one is phosphatidylinositol. If you have again the serine, phosphatidylserine. If you have the ethylamine, phosphatidyl. If it's choline, phosphatidylcholine. So other examples of phosphoglycerides, you have here again the general structure. You have the ester, you have the fatty acids bonded, which is unsaturated phosphate, and the X. Your X again can be like just hydrogen. Then you can have your phosphatidic acid. You have your, you have ethanolamine, you have phosphatidylethylamine, choline, phosphatidylcholine. This is like your lecithin in the egg yolk. Serine, phosphatidyl-serine, you have here, okay, remember the structure is serine. Or if you have another glycerol, you have your phosphatidylglycerol here. So for phosphatidic acid, no, what's being added here is just hydrogen, no. So simply as phosphoglyceride or simply as phospholipid. The alcohol is sterified to phosphate group. And isa pa. Glycerol, no? If your X is glycerol, you have your phosphatidyl-glycerol. If your X is inositol, 4,5-biphosphate. So you have your inositol here, kani OH, OH, up, OH na siya supposedly, But when the hydrogen is being replaced with phosphate group, then you have your 4,5-biphosphate. Okay, so carbon 1, 2, 3, 4, and carbon 5, there is a phosphate. So duha ka phosphate, biphosphate. So you have phosphatidylinositol because this is inositol. But the inositol is being substituted with 2 phosphate groups. So phosphatidylinositol 4,5-biphosphate. You have here phosphatidylglycerol. You have here kani H2OH. Mura siya na-repeat, no? And what you see here is the glycerol again, glycerol CH2. On the phosphate group, you have here CH2O, you have fatty acids here bonded. The phosphatidyl the name is cardiolipin. So more siya na-repeat imuhang phospholipid, but with the glycerol added there, the triglyceride rather, you can see here, but is terrified on this. Okay. Isa pa. So the similarities in shape and molecular structure of phosphatidylcholine and sphingomyelin. So there is another class of phospholipid, which is sphingomyelin, but there is a difference in their structure. So earlier talked about the phosphatidylcholine. Okay. If one of our tanawin lang ninyo ng pangitaan lang ninyo ang glycerol, H2OO. Okay, so O, this one, the glycerol is sterified with fatty acids, and the third carbon is, you have the phosphate P, double bond OOOO, oxygen, and one of the oxygen is bonded another group. This is a choline, this is phosphatidylcholine. Now, if you look at on the structure, naani siya, naani si choline, no? You have your CH2, CH, mura siya old glycerol, right? Pero mura pug dili, you don't see the esterified one, but instead you see an amide here, okay, with our NH instead of OH. O here, you have NH, and COOH here, you have your hydrocarbon here. So here you maintain the phosphate group with the choline here. And this one is a kind of sphingomyelin. It's a sphingosine. No? So we'll look at on the structure of another kind of phospholipid, which is the sphingomyelin. No? In the sphingomyelin, instead of a glycerol, you actually have the sphingosine. So what is the sphingosine? The sphingosine actually is more siya glycerol. Tanawan niyo ng first 1, 2, 3 carbon na siya OH, but the second carbon is na siya NH2. And on the third carbon, instead of OH, na siya carbon change. It's 13, 14, 15, 16, no? 13, 12 plus 1, 13, 14, 13, 14, 15 here. Okay, and there is a double bond near the third carbon. So the sphingomyelin contains sphingosine instead of glycerol. And what you can see in the structure, you have here the sphingosine. On the second carbon, you have the NH2. On the first one, you have the OH. And you have here, this is the OH, where you can have your substituted with another group. This is the hydrogen that's being substituted, and the NH, where the hydrogen is being replaced with another group. And this one na maintain rin siya. So you have your OH on the third carbon, and here you have carbon 1, 2, 2 plus 13, 15. So you have 2, 13, 14, 15. On the second carbon, where it is attached to your third carbon ng AOH, no? Nakay double bond. Okay, so tanaw na yung fluorescent structure. So this is the kanimo ning sphingosine, including this one, but the first OH and the first carbon with the OH is being substituted. The OH hydrogen is being substituted with another group, and the hydrogen in the NH2 is substituted with another, your fatty acid you have here. So this is the structure of the general structure of your sphingolipid. And one example of the sphingolipids I've mentioned is you have the sphingomyelin. Okay, so kita na po natin structure para maka-identify ta, no? Is it lipid or what's it? What kind of biomolecule is this one? So na-phosphates, makaingon ta, this is already a most probably is phospholipid with so many carbons attached to it. But you have to see asang a part, no? Asang a part, phospholipid. Sphingomyelin, not sphingomyelin. Sphingolipid. You can see here, you have the NH, you have the CH2, and this is where the substitution occurs. Kani, this 1, 2, 3, first 3 carbons, you have the NH, you have here, and you have the CHOH, where this is where the 15 carbons being added, 12, 13, 14, 15, with the double bond here. And on the first OH, no, ang na-replace sa iyaha is a phospholipid with a choline here, choline group. Okay, so this is what you call as a sphingomyelin here. The combination of a fatty acid with sphingosine is called the ceramide, kani. Fatty acids, this is where your fatty acid is, and the sphingosine, this is where your sphingosine is. Your fatty acid is bonded to the NH. Instead of seeing sidyl bond O ester, what you can see in the fatty acid part is being esterified. It is nahimo siyang like forming an amide, you can see here. So because many of these compounds are also found in cerobrosides. The ceramide part of the complex may contain different fatty acids. You have here the R contains, you may have different R here, different R groups of fatty acids. So stearic acid, for example, occurs mainly in sphingomyelin structure. Okay. The sphingomyelins are the most important lipids in the myelin sheets of nerve cells and are associated with disease such as multiple sclerosis. Is anyone here who is having sclerosis? No, it's actually some disease in the nerves, right? So sphingolipids are not randomly distributed in membranes. In viral membranes, for example, most of the sphingomyelin appears on the inside of the membrane. So there was this guy called Johann Tudishom who discovered the sphingolipids and named this brain lipids after a monster, Greek mythology, the Sphinx. So the Sphinx. Now, I don't know if you're into Greek mythology. Anyone is into Greek mythology? Naba? Wala. We'll have to read Greek mythology. Part woman and part winged lion. Remember the Odyssey? I think it was in the Odyssey of Koan. So the Sphinx devoured all who could not provide the correct answer to her riddles, and Sphingolipids appeared to Tushidom as part of a dangerous riddle of the brain. Okay, so any, so sphingomyelin is part of the important lipid smash sheets of nerve cells. So any defect on that would lead to multiple sclerosis. The sphingomyelins resembles phosphatidylcholine in their general structures. That's what we have already said earlier. We have shown on the structure the difference between sphingomyelins and phosphatidylcholine, like katong lecithin, no? In sphingomyelins, we're using sphingosine. In phosphatidylcholines, we have the glycerol. So in their general properties, and you have the three-dimensional structure in having no net charge on their head group. Sphingomyelins are also present in plasma membranes of animals and are especially prominent in myelin, a membrane sheet that surrounds and insulates the axons of some neurons. Okay, so our neurons actually, our neurons now have the axons, and it's the one that insulates us now when we're having like signal transduction in between nerves. And you have there the sphingomyelins that insulates the axons of the neurons, and it facilitates the rapid transmission of nerve impulses. One of the reasons you can see is that sphingomyelins actually are, you have the hydrophobic part, but there is a part which is polar, very polar, and with that can easily transmit signals, no, from one end of the nerve to the other end of the nerve. Can have the nerve impulses. Okay, so again, this is your sphingomyelin. You have here, we have the choline part here, and you have your fatty acids here. Okay. So the sphingolipids, we have the sphingolipids, second classification. You have the sphingolipids. You have the sphingosine, again in the structure. And you have the glycolic, no? You have your fatty acids in the second, the sphingosine, and you have your X. So if your X is phosphocholine, like siya, phosphocholine, okay, then you have your phosphocholine. Sphingomyelin, rather, you have your sphingomyelin. If you have your phosphocholine, if you have your glucose, then you have the neutral glycolipids, glucosyl cerobrosides. Okay, so you just have to take note on this truncon. This is our phospholipids. Okay, another part is the glycolipids. Another classification is the glycolipids are complex lipids that contains carbohydrates and ceramides. And some ng ceramides, katubitawang na ay sphingosine. Okay, and nakay NH, no? NH, where you can have your fatty acids there. So one group, the cerobrosides, consists of ceramide mono or oligosaccharides. In other groups, you have the gangliosides, contains more complex carbohydrate structures. You have here. So in cerobrosides, asama na cerobrosides, kani, tanaw na to. This is your ceramide. Diba remember, paintaon na to carbon nga na OH. Kani, carbon nga na O, but ilisan si X, you have the X, and you have the second carbon, which is NH2 supposedly, but ma-replace one of the hydrogens in H2 is ma-replace with another group, your fatty acids here, and then the other one, the carbon OH, is actually you have your long chain. This is constant here. So this is yours, kaning structure, kamandiri. You have your ceramide, can have here. Again, you have your fatty acids here and you have your glucose here. So other groups such as gangliosides contain a more complex carbohydrate structure. So here is your carbohydrate structure attached to the third carbon. In cerobrosides, the fatty acids of the ceramide part may contain either 18 carbon or 24 carbon chains. Kani, you may have 18 to 24 carbon chains. And the later form is found only in the complex lipid. So pag taas-taas ng carbon, you can find that in a more complex lipid. A glucose or a galactose carbohydrate unit form a beta glycosidic bond with the ceramide portion. Kani. So you can see here your glucose. So down, up, down, up. Remember, nasa H2 na siya. You can have here, you have your up. So it's a beta glycosidic bond, O bond there. Here ceramide, with the ceramide portion of the molecule. And the cerobrosides just contains your ceramide mono or oligosaccharides. The cerobrosides occur primarily in the brain. So the cerobrosides occur primarily in the brain, accounting for 7% of the brain's dry weight and at nerve impulses. So you can now know that our brain is composed primarily of our glycolipids or cerobrosides. Now, the cerobrosides are sugar linked to ceramide kani. Cerobroside sugar linked to ceramide kani because this is our ceramide. This is our sugar part. Depende na kung saan sugar. Those with galactose are characteristically found in the plasma membrane of cells in neural tissue, and those with glucose in the plasma membrane in non-neural tissue. So imagine, no, makabantay na mo nga natin ang mga just a discussion, no? Why gonna bitong people who were not eating for a lot of days, especially pag nasa sila gunahon, no, more sila mawadan sa ilahang panghunahuna, okay? Because you need to have glucose in the neural and the tissues of your brain, okay? In the non-neural and the neural tissues of your brain. So katong in the neural tissue of the brain, the plasma membranes of cells in neural tissues, you can find galactose, but sa mga non-neural tissues, you can find glucose. In the example here, we have glucose, but changing one supposition of the OH, you can have your galactose, okay? So example, you can have your galactose cerobroside entirely found in the cell membrane of the brain. And you know that galactose can find that sa milk, sa mga milk, right? So when you are young, it's important if you are soon to be mothers, if you want to have a child in the future, make sure that you feed your child with milk because it contains a lot of galactose, okay? And it will be used for, it will certainly be used for brain development, especially sa mga bata. Okay, so another one, you have the gangliosides. So it's considered as a glycolipids having no phosphate groups. Gangliosides, the most complex sphingolipids, have the oligosaccharides as their polar head and groups and one or more residues of N-acetyl-neuramic acid or Neu5-AC. N-acetyl-neuramic acid. Sa mana kani. Mura siya og na sugar, no? But one of the mura siya glucose or mura siya og monosaccharide, not sa ring. One of the carbons, you have this structure. You have CH, is OH, and then you have here, and then you have your amide group here. You have here. This is your N-acetyl-neuramic acid or the sialic acid. Sialic acid gives gangliosides the negative charge pH that distinguishes them from globosides. And if you can isolate this gangliosides from nerve tissue and other animal tissues. So again, in this gangliosides, they are complex sphingolipids, but you can see here, you don't see any phosphate group in the structure. Okay, so also you can have lactoceramide, okay, depending kung unsang imuhang X nga ma-add sa imuhang formula. Saan na imong formula? Kani, ayaw mo kalimot. Kaning atong X, now we're referring to the X here. You have your sphingosine rather. And sa imong X, pag naaga ni phosphate, usually you can see here phosphate group for Sphingomyelin, but your X can also not contain any phosphate, no? Like you can only have your glucose or any other sugars, you can have your glycolipids. And if your X is kani, can have glucose, galactose, or you can have your complex oligosaccharide glucose, galactose, and you have this structure Neu5-AC kani, the structure, or you have your another structure here, you know. You have the ganglioside. So what is the importance of sphingolipids? Or partly when Johann Tukidum discovered sphingolipids in around 20th century, he had no idea why they were present. That's why the names derived from Sphinx. No, actually derived the Sphinx again from the Greek mythology. We now know that many are involved in cell surface recognition. And one well-known function of sphingolipid is that they function as cell surface antigens defining the various blood types. And I was just talking earlier about katong sa brain, where you can find your sphingolipids. But one importance is that this one can also function as cell surface antigens, which defines our blood types. Now, for example, you have the ceramides, sphingosine, and you have the second carbon, you have your fatty acids, and depending on the antigen, whether it's glucose, galactose, ketone, complex sugar-like structure, N-acetyl, galactose N-acetyl, you have galactose, and you have so many. This is the fructose, and then you have another ring here. Okay, so depending on the ring that's being bonded to the third carbon here, depending on the structure, you can have this different antigen that would differentiate the different blood types. This is the antigen that will bind to any other antibodies. Okay, you have here. So for example, you have O, A, B antigen. That's what they have discovered on the sphingolipids. Many are involved in cell surface recognition sites, and one well-known function is that they function again as surface antigens, where we define our blood types. I hope that you know your blood types. And the human blood groups are determined in part with the type of sugars located in the head group. So in the ABO system, blood type ABO, blood type, the determining gene is located. So this is very high. Okay, so if you reach into the DNA topic or in your biology, you can have the determining gene gene for the blood type. It can encode different antigen. In the process, if the gene encodes a protein that transfers an N-acetylgalactosinamide group, this corresponds to an A antigen. You have here N-acetylgalactosamine, N-acetylgalactosamine. If gene encodes a protein that transfers galactose group, you have the galactose group, then you can have the B antigen. So depending on the protein on the gene that encodes a protein and a protein that transports a galactose. In the next chapters, in mga 2 chapters, I will be discussing enzymes first, and after that, we'll be discussing about nucleic acids, and hopefully we can arrive into the replication, transcription, translation part, where you can have your proteins. Once you get your proteins, the proteins, of course, you've learned in your protein functions of the proteins. It can form as a transport protein, it can form as a catalyst, can be used as a catalyst. Proteins would have a different function. So if, for example, a gene encodes a protein and the function of the protein is just for transport, for example, in this case, will transport galactose group, so inyong blood would be defined like B. So anyway, that's just an application. Don't bother so much of this. Just an application of your sphingolipids, where the differences on the X, on the complex X, complex oligosaccharide that you can see would actually define on the blood type that you have. So in humans, at least 60 different sphingolipids have been identified in cellular membranes, and many of these are especially prominent in the plasma membrane of neurons, and some are clearly recognition sites on the cell surface, but a specific function for only a few sphingolipids have been discovered thus far. The carbohydrates moieties of certain sphingolipids define the human blood groups and therefore determine the blood types of the individuals. So other rules, some glycolipids may bind bacterial toxins as well as bacterial cells to animal membranes. And because of that binding, we can have actually immunological response. Examples, the toxins that cause cholera, tetanus, and diphtheria bind the glycolipid cell membrane receptors, and if it binds, no, there is so much or other system or the defense mechanism of the body may recognize that one and would cause our body to have inflammation effect. Lysosomal storage disease is caused by the hereditary deficiency of an enzyme required